for tuning in to Planetterrian Daily, episode 126. It's July 20th, 2026. Here's your science and health briefing. AI analysis shows climate shifts drove repeated bursts of bird evolution across Earth's history. A study mapped gene expression and protein changes in muscle after sustained physical activity. More than half of age-associated molecular features reversed or attenuated with exercise. The remaining unchanged markers highlight pathways less responsive to exercise alone. Remember, the show covered aging biology on EP 125. Today's news moves that forward because it identifies which specific molecular features of muscle aging respond to exercise. Now, shifting to a very different area of research. University of Michigan researchers applied an AI tool to passeriform species across geological time. The analysis revealed rapid evolutionary bursts that repeatedly aligned with major climate transitions. The pattern emerged from comparing diversification rates against paleoclimate records. This approach allows direct testing of environmental drivers on speciation without relying solely on fossil counts. Next steps include extending the method to additional avian clades. On a different note, EMBL Hamburg and Leibniz FMP teams developed a customized workflow to track protein contacts inside living infected cells. The method captured how the virus systematically alters host interaction networks at multiple stages of replication. Direct observation inside intact cells preserved spatial context lost in traditional lysate approaches. The resulting map shows specific rewiring events tied to viral protein functions. Follow-up work will test whether blocking identified host targets limits viral spread. Next up, the Cybra suite connects reference brain atlases with heterogeneous datasets stored across separate repositories. It enables joint exploration, analysis, and reuse of large, multimodal brain data. The tool handles differences in resolution and acquisition methods without requiring data centralization. Developers report that the system supports queries spanning molecular to network scales. Continued integration of new datasets is planned. A compact fluorescence analytical instrument has been developed to perform molecular assays outside laboratory settings. The design targets applications such as symptom tracking and initial illness screening by patients themselves. Miniaturization maintains sensitivity comparable to larger benchtop systems for selected targets. Developers highlight reduced need for sample transport and faster turnaround. Validation across additional assay types is underway. The instrument fits in a form factor comparable to consumer earbuds. Early prototypes have already completed internal bench testing for core assay panels. Participants reporting the hum underwent hearing tests and ear sound measurements. Neither exceptional low-frequency sensitivity nor spontaneous otoacoustic emissions accounted for most cases. The data instead point toward low-frequency tinnitus perceived as external. The finding narrows diagnostic approaches for clinicians encountering similar reports. Larger population surveys are needed to confirm prevalence. Follow-up protocols now prioritize tinnitus matching procedures over sensitivity thresholds. From targeted human interventions to broader evolutionary responses to environmental change. Next, we look at how climate has repeatedly shaped bird diversification. Exercise erased more than half the molecular signature of muscle aging in a recent mapping study. The work distinguishes which age-related gene expression and protein changes respond to physical activity from those that persist. This distinction matters for designing combined interventions that address both exercise-sensitive and exercise-resistant features of sarcopenia. Listeners should watch for human cohort studies that test whether the same molecular split appears with different training protocols. From planetary scale environmental drivers to the molecular interactions inside a single infected cell. We turn to new work on influenza. Researchers synthesized cosmic dust analogs inside glass tubes under space-like conditions. The resulting particles contain complex carbon-rich molecules built from elements essential to life. Infrared spectra from the lab samples match signals detected in actual interstellar material. The work isolates how organic chemistry proceeds around stars before incorporation into comets or meteorites. Further experiments will vary temperature and radiation parameters. From cellular maps to whole brain data integration, the next tool connects reference atlases with multimodal datasets. A new study identifies an unexpected route for seeds to synthesize and store a key milk protein. The mechanism allows accumulation without disrupting normal seed development. This finding addresses a bottleneck in efforts to generate animal-free dairy proteins at agricultural scale. The work focused on one protein but suggests broader applicability across additional milk components. Field trials of engineered lines are the next reported milestone. From mapping healthy brain structure to mapping the origins of its building blocks, we move to laboratory recreations of cosmic chemistry. Researchers generated red blood cell-like cells from canine-induced pluripotent stem cells to address transfusion compatibility challenges in veterinary medicine. Dogs possess multiple blood types, making donor matching difficult with current reliance on live animal donations. The approach demonstrates a potential route to produce typed blood cells in vitro. Compatibility testing and scalability remain key hurdles before clinical use in dogs. From chemistry in space to chemistry inside seeds, we next examine an unexpected route to animal-free dairy proteins. Surveys from the 1960s had flagged a possible deep-water reef but assumed it was dead. Modern sonar combined with the National Geographic Deep-Sea Camera System revealed an apparently healthy reef structure. The find contradicts earlier assumptions about the region's benthic habitats. The reef supports diverse marine life despite its location in an area long considered unsuitable. Additional mapping will determine its full extent and species composition. From engineered plants to engineered blood cells, the next study explores stem cell-derived transfusions for dogs. Imperial College London and Taipei Zoo researchers recorded male dragonfly maneuvers during territorial disputes. The fights consist of repeated sequences of chases, turns, and positional adjustments rather than random flight. These patterns appear to serve both territory defense and mate attraction. Video analysis quantified the frequency of specific tactics across multiple encounters. The team will next examine whether females use fight outcomes in mate choice. From cell production to habitat discovery, underwater drones have now confirmed a living reef where none was expected. Researchers showed that controlled energy leakage from quantum systems can generate entanglement between qubits. The method exploits environmental coupling that is normally viewed as a source of decoherence. By tuning leakage rates, the team produced entangled states across channels that would otherwise destroy coherence. The approach offers an alternative route when standard transmission methods face high noise. Further tests will assess scalability in larger qubit arrays. An aluminum igloo-style platform left France to drift with polar ice for extended observation. The station is designed to operate in extreme conditions while collecting continuous environmental data. Its mobile design allows sampling across changing ice regimes without fixed infrastructure. The mission duration will span multiple seasons of ice movement. Data will feed into models of Arctic system dynamics. The platform carries modular sensor arrays that can be swapped during resupply windows. Initial drift trajectories have already been logged by satellite beacons. Harvard Kennedy School researchers examined how shared team allegiance creates cross-group connections. The analysis links fandom participation to measurable improvements in social cohesion metrics. Effects appear strongest when fans engage in sustained group activities rather than solitary viewing. The study proposes fandom as one underused lever for addressing polarization. Larger intervention trials are planned. Surveys captured both online and in-person fan communities across multiple leagues. Baseline measures of perceived social distance were collected before and after season-long engagement. Before we go, keep an eye on pilot programs testing streamlined mosquito surveillance protocols that integrate trapping data with climate records. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at Nara Network. Links in the show notes. And before you go, this show is part of the Nara Network, a family of daily podcasts on tech, science, and markets, each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. Models and Agents, your daily briefing on AI models and agents. Find every show free at naranetwork.com. Want the written briefing with source links? Each show keeps a summaries page on naranetwork.com. Scannable, dated, and free. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.